Well, thank you very much, Mr. Wallenberg. Thank you to the foundation for having me here. And I assume the mic and the technical is going okay? Lovely. It's really an honor to be here to help celebrate Jerry Tuscan's great award. Jerry and I have known each other for about 40 years. That's about the span of our career, thus the 44 plus decades of rapid change. So what I'm going to do today, very simple. I'm going to try to look at some of the highlights of change to give you a sense of really how awesome, how amazing it's been, a little bit of context to understand some of the more technical talks that will come up, and then talk a little bit about the challenges of using this amazing genomic and molecular science out in society for good. So we'll end with that and then come back briefly to talk about our honoree, Dr. Tuscan. So this is from a paper that Wout Berrien, who's here somewhere, and he actually was using BioRender to get this done, but we worked on it together. I think gives a nice overview that'll help you, I think, put everything to come in context. So when we do tree biotech or genetics, generally we're using natural genetic diversity. We may amplify it by making hybrids and putting them in unique environments. And then we're putting them in the field in some structured way so we can see their genetic value versus their phenotypic value, their environmental influences. And that's mostly how we do breeding, right? We see, are the trees growing fast? Are they disease resistant? Do they have good wood properties and so on and so forth? And then increasingly today, and you'll hear some great talks about this coming up, we can use the genome. We can put tens of thousands of markers throughout the genome and use that as an index. We train that empirically, meaning we go to those same field trials and correlate the genomic information with the phenotypes. So it's not based on knowledge of the genes. It's based on just having so many markers that we can characterize the genome in general. So, and this technology, because of the sequencing revolutions that have happened, computation, that is pretty mature technology. Anybody can do it today. It's really a matter of economics. So that's very exciting. That's the left side of the panel in front of you. On the right side of the panel is what I'm calling functional biotechnology. It starts with knowledge of the genes and the pathways and what they do so we can tweak them in an informed manner. We can make a new kind of gene that's expressed a little differently, put it in. It can come from the same species or a different species, or we can edit the gene, change some bases to make it do something we want it to do. And that, and you see this ideal of these things coming together. This is like what biotech companies do. They're doing conventional breeding with genomics, but they're also adding in these particular genes for particular traits. And in forestry, really can't do that yet. It's very, very rare worldwide to be able to have the functional side come in. So we'll talk about this cleavage and what we might do about it as we go. So overview, where have we come in 40 plus years? Obviously, genomic methods and diversity tools. I'll talk about the diversity tools in a little bit of detail. We're just coming online. Prior to this era, we really, apart from quantitative variation in the size of trees and what they looked like, we didn't really know what diversity was. We couldn't measure it. We couldn't quantify it. It was a very, very rough science. And then this thing happened with molecular markers that I'm going to tell you a little bit about in detail. And then that led to many more markers, eventually to genome sequencing. So I'll tell you about that paradigm change in a minute. And then, of course, and I think you'll hear more about this from Jerry and others, we have this sequencing explosion of capability, which is both on the laboratory end and the computational end that Jerry and his team helped pioneer. And then we had this thing I'm calling genome-fueled molecular physiology or functional gene biology. I've also called it in some slides molecular-enabled biotechnology. This is the functional side I showed you earlier, where we're using knowledge, scientific knowledge, to make intentional genetic changes, and tell you a little bit about how exciting that is. So an example of where we were at, the first IUFRO meeting on tree biotechnology. I think it was 1984, maybe 1985. Jean-Ray, you know this better than I do. But somewhere way back there at the start of my career and Jerry's career, I was at a meeting in Ohio, And the state of genome analysis was represented by this. These are Cott curves. We're taking DNA, we're boiling it to separate the two strands, and then letting it slowly come back together and looking at it. And you see that curve up top, it has this very interesting, strange behavior. And that really tells you that there's highly repetitive DNA in the genome, different fractions of it, they're modeled down below. But that was really the state of genome science. This very coarse, it's almost like the Dark Ages compared to the Renaissance. That's where we were at the start of my career and Jerry's. Then what I'm going to do here is I'm going to talk about now this development of markers so we can really see genetic diversity and use it. And there's a paper that I found by some very well-known population geneticists. You can see their names right there, who looked back. And it really helps put in context how important what their work was. And I have some quotes from that paper. So a 1966 genetic paper was a landmark in the study of genome-wide levels of variability, using gel electrophoresis of enzymes and proteins to study variation. It started in Drosophila, the fruit fly, and they used a sort of random set of loci that hadn't been chosen for any reason. So it was, at the time, sort of as close to a random look at diversity as they could get. And this is what one of those things looks like. These are protein extracts put in an electric field, and the proteins are migrating at different speeds because of differences in charge, differences in their amino acid composition. So it represents genetic variability almost at the gene level. And we can interpret those in terms of gene frequencies and so on and so forth. But going on with that paper, together with a study that interesting was going on at the same time in humans doing the same thing, they found that they could find a relatively unbiased picture of genetic variability, and there was extensive diversity, much more than anybody thought. And there's great discussion and debate that continues to this day about, is that diversity mostly functional? Is it mostly just mutation, sort of noise, or some mix thereof? A lot of discussion in the field at that point. And then finally, and most interestingly for us, the paper stimulated a large research program that found similar things in every organism pretty much that was studied. And then at the same time, that stimulated a lot of theoretical development in terms of how, whoops, let's see if I broke it. Nope, still seems to be intact. In terms of genetic drift, evolution, phylogenetics, so there's a flowering of these computational software tools allowing us to interpret it. But again, these studies were so widespread, and they were going on in the 70s and 80s when my career started and Jerry started, and we both engaged in these marker studies, that we could look back at thousands, tens of thousands of species, animals, plants, microbes, and look at their levels of diversity and relatedness. And these, of course, this led eventually to genome markers, as you'll hear. So what we wanted is something closer to the actual genome than those protein variants were giving us. And so don't worry, I'm not going to go through all of these acronyms. Geneticists love these things, by the way. This is how we talk, but I'm going to spare you. This is to show you that what's happened from over time, over the last 40 years, to go from these protein variants to now being able to have tens of thousands of markers throughout the genome and full genome sequences. So one of them was called RAPID, R-A-P-D, randomly amplified polymorphic DNA. It was a brilliant, I remember seeing the talk from some scientists at DuPont where you randomly amplify parts of the DNA. And I should step back and say, I don't know how many of you in this audience know PCR, polymerase chain reaction, but when I started and when Jerry started, there was no such a thing. Whenever I tell my students we were doing molecular biology without PCR, they gasp because everything was so much harder. But PCR also led to these amazing markers like RAPIDs. And so there was a variety of papers. I'm just going to mention one by our honoree, being able to use these markers to very quickly see if you're part of a clone of Aspen or you're a seedling cohort, which is very important to their regeneration and demography. And this is what they look like. There's these bands on gels. There's really too much information, but some of them behave nicely. You see the arrows here where you can interpret their genetics. And so work like this happened in many different laboratories, and we got enough of these markers that we could almost saturate genomes with them. And we started thinking, wow, for the first time, we have lots and lots of markers almost throughout genomes. what can we do with them? Can we use them for practical purposes? And so the question, could these markers be useful in tree breeding? And so our honoree, Dr. Tuscan, put together a symposium that had an enormous impact on me. He knew that I was a curmudgeon and tended to be skeptical about all these fancy new technologies. So he said, Steve, why don't you express your doubts? And I did it and wrote a paper about it. And it was fairly influential at the time, but it was based on this sort of old marker technology. And so the conclusions, which are mostly that this stuff won't work and won't be affordable, turned out to be completely wrong because we had such breakthroughs in genomic and computing technology. And you'll hear about that from several of our speakers today who are now using it very productively. But I thank Jerry for bringing me together with a lot of other colleagues to think this through. And I've always noticed he's kind of like Abraham Lincoln in the United States. He welcomes his rivals because he wants to make the best choices. He's not afraid to hear divergent viewpoints. Thank you, Jerry. So, and then this thing I'm calling molecular empowered biotechnology. This is hard to describe, but genomics is not just sequences. There are all these other things. Transcriptomes, meaning the expression of all the genes in an organism, in a cell, at different stages of the life cycle. Metabolomes, the different metabolites that are produced. Epigenomes, methylation and DNA modification, is very important. So all of this, and then phenomes, looking at the phenotype, and I think you'll hear a little bit about that from Jerry today. Whether it be drones in the field or whether it be hyperspectral imaging in the laboratory or in the field, we can really look into the organism with precision and speed unthinkable previously. And then I don't have time to talk about all of this, but being able to do gene transfer is critical. So modifying agrobacterium, the main gene transfer agent, because that's how, when you see a gene statistically in one of these fancy computer analyses, you really don't know what it's doing with biological certainty until you perturb it in isolation of all the other genes and put it back in an organism. So having transformation is critical. And then also having an organism like Arabidopsis, which is a little plant that completes its life cycle in about 6 weeks. It was the first genome sequence determined. And when we look at these genomes, they're just A, T's, G's, and C's. To make sense out of it, you need to look at what we know about the model plant, Arabidopsis. So everything we do in this area of genomic-empowered biotechnology goes back to Arabidopsis. And luckily, the world had the wisdom to invest in the Arabidopsis genome very early on, and that helps all of us in all the crops, including trees. So because of all this, we really have an abundance of options. We know lots of the genes involved in stress tolerance, lots of the genes involved in lignin modification. The question is really where to start. There's so many opportunities, how to make choices, how to deploy them in the field, and then as we're here, will society let you deploy them? What techniques are acceptable and what are not? So let's move into the second part of this talk, which is really about some of the challenges. So we have extraordinary genetic diversity in forest trees. In fact, those Allozyme surveys I mentioned earlier showed that trees are among the most diverse organisms on Earth, bar none. It's extraordinary levels of diversity. And so one of the things that we can do, and Jerry helped pioneer, is have extremely dense markers that can allow us to really, better than about anywhere else, recognize which genes are doing the action. So very powerful. Nonetheless, and it may seem a little paradoxical, when we dissect traits, we tend to see that the major traits that are important in forestry, the complicated traits like yield and wood density, are controlled by numerous genes that have small effect. So Dario is going to talk about that a little bit in the lecture he's going to give this morning online. And that means this powerful technique, there you go, another acronym, sorry about that, GWAS, Genome-Wide Association Studies. That's when you read in the newspaper about a new gene for a disease, a hereditary disease in humans, it's usually isolated through that method. Scanning genomes in tens of thousands of people to look for the sources. Well, our experiments tend not to be that big. We can't afford the gigantic budgets you have in biomedical science. And the ones we've done pretty much for complicated traits, suggests lots and lots of genes of small effect, and you'll see that from Dario. So it's very hard to use the findings from GWAS directly to accelerate tree breeding. So the genes of most importance, if we want to have genes that have big effects, they need to come from other places, not from GWAS. They can be new modes of action from other species, and that can be very, very powerful. And of course, that's had a revolutionary impact on agricultural biotechnology. Or they'll come from these function-based molecular-empowered biotechnology studies. And now GWAS can really be helpful here. It may not be the individual genes, but seeing the pattern of what genes are important tell us what pathways are important, and then we can tweak the pathway. I know this is getting into the weeds a little bit. So I just want to make it clear that GWAS is not a useless technique. It's a very valuable technique. But the inferences are not so much about the individual genes, but about the biological findings and how you use it to inform biotech. So fortunately, you know, and you'll hear about this from our speakers, including from Jerry coming up shortly, there's a plethora of findings and exciting things happening from this genome-empowered functional biotechnology. So they come from many, many different places. I've just named a few of them. This is putting it all together to figure out really how organisms function and how we can tweak them. They involve every trait of interest, of commercial interest, growth enhancement, wood quality, pest and stress resistance. And the real problem is that not that we have a shortage of ideas and actual plants, even in greenhouses, looking attractive, it's really field verification. And you all may know here that plant breeders don't breed on a computer or in a greenhouse because the performance of plants in the real world is different than the performance of plants in cultured, highly controlled environments. So really it's based on what happens in the field over many years and many different environments. So we really need to be doing that to use this information. The problem is that it's hard. So why do we have this division between, I'd say, genome marker-based breeding and biotech and the functional side? So one thing is the gene function-informed transformation and editing so far tends to focus on simpler traits. It really needs to. And the amazingly powerful work in lignin modification in poplar, and I think John and Wood are here, the Marcus Wallenberg winners last year, is an example of how beautiful this can be. But the simpler traits and lignin is very much a subset of the problems we have in forestry, not the top priority traits. And as far as I know, and please correct me if I'm wrong, these have not yet been integrated into breeding programs. We have some lovely field trials that are very encouraging. But this is one of the best examples that it can work if we get the other stuff right. One thing that's close to my heart in terms of the research in my lab now is transformation as an obstacle. It's very difficult to put the genes in and modify them or edit them at a scale relevant to breeding, which involves many, many genotypes, always changing, always moving forward. And then there's the social side, regulations that restrict where you can grow at all, and then things like green certification that are throughout forestry that say if you use those techniques, you can't have our stamp on your products. We'll decertify the entire company, in fact. So we have these two major social issues. So the world is slowly moving toward relaxing some of them, and I guess it's a hot discussion point here in Europe, but it's completely inadequate to the opportunity, and it's not clear it's really going to make a difference. But there is a little bit of light, and you have to take what you can get. So, and I see I'm running a little low on time, so I'm going to move a little faster here. So this is something close to my heart. So over my career, I've worked with a number of colleagues to write about this constraint, these constraints of regulation and market barriers and ways to get around them that are based on science. So, and this slide, I didn't animate it because I didn't expect to have time to talk about it, but I'd love to. This is great stuff to talk about over a beer, by the way. You know, why do we have this split? Why do we have scientists saying one thing and certain scientists versus other scientists? And you can just read down, there's a lot of very fundamental issues that are underpinning this. So it's hard. it's a hard issue. There's not going to be one answer. So finally, I want to end by coming back to Dr. Tuscan here, our honoree. Like myself, he's been very involved in every step of this evolution over the years, trying these new techniques and so forth, and publishing excellent science. And then something happened. Something wonderful happened. And you'll see the public face of it in a minute. Before I did, Jerry saw the power of genomics and what it would do to biology. So he used his platform at the Department of Energy to rally lots and lots of scientists to sort of start paying attention to genomics, and I think he'll talk about that. And there have been just untold scientific insights, including biotech insights as a result of that. And so to me, it's the genome paper he published that marks the big transition. So it really, so this is the paper published in Science in 2006. It's got more than 100 authors. It's been cited over 5,000 times. It is a big deal in all of biology. And I was actually a peer reviewer. I've told Jerry that. At the time, I didn't let him know. I was a good anonymous peer reviewer. But I'd never read a paper like this before where you had such an interdisciplinary effort, computational scientists, all kinds of physiological scientists coming together to do this genome and then interpret this genome. So very, very exciting, and really has been part of this revolution we're living through that's empowering this functional biotechnology. So to finish up, obviously no one's going to argue with this. It's amazing where we've come in 40 years, and it's going strong. All the things that promised are happening, providing new opportunities for biotech. But we have this cleavage of what we can do in breeding and what we can do in functional biology as a result of the polygenic nature of traits, transformation limitations, and strong social obstacles. So there's no question either that this guy over here, your honoree, has been a leader at every step of the way. So it's a real pleasure to celebrate you today, Jerry. Thank you very much.